Assalamu alaikum. Today we discuss about the nitrogen fixation. First of all, we talk about the discovery of the nitrogen fixation. So nitrogen fixation was discovered by Dutch microbiologist Martinez. Dutch microbiologist Martinez. So first of all, we talk about the introduction. The nitrogen is an essential component of DNA and RNA and also the protein. Although majority of the air we breathe is nitrogen, our atmosphere contains 78% nitrogen in our atmosphere. So two atom nitrogen molecule, this two atom nitrogen molecule, when break apart, that nitrogen can combine with other atoms like oxygen or hydrogen. So nitrogen gets fixed when it is combined with oxygen or hydrogen. So when the nitrogen degrade and break down, they combine with other elements like hydrogen or oxygen. So when it can bind to that, that can fix the nitrogen. And so now we talk about the definition of the nitrogen fixation. Nitrogen fixation the conversion of the molecular nitrogen into ammonia or nitrates is called nitrogen fixation. So what happened? Nitrogen molecule convert into ammonia or nitrates. It can convert into nitrates or ammonia from the nitrogen. So this process is known as nitrogen fixation. This is known as nitrogen fixation. So there are three ways to fix nitrogen. One way by atmospheric nitrogen fixation, and second is the industrial nitrogen fixation, and the third is the biological nitrogen fixation. So these are the three ways to fix the nitrogen. So today we'll discuss about the biological nitrogen fixation. Biological nitrogen fixation. So the biological nitrogen fixation, this is a type of the nitrogen fixation, which is carried out by the bacteria, is called biological nitrogen fixation. This fixation by using the bacteria. This is nitrogen fixation carried out by the bacteria, so that are known as the biological nitrogen fixation. Then these bacteria convert nitrogen into ammonia, and they convert nitrogen into ammonia or nitrates. So the nitrogen fixing bacteria are present in two states. They are present in two states. One are the free living state, and second is the symbiotic state. So these are the two states in which the nitrogen fixing bacteria, one are the free living state and other the symbiotic state. So for example, we take an example like nostoc. Nostoc present in free living as well as symbiotic state. It can living in both states, free living and symbiotic state. Like most blue-green algae, nostoc contain two pigment. They contain two pigment. One are the blue phytocyanin, and second are the red phytoerythrin. So and also as well as chlorophyll, as well as it contains chlorophyll. So what happened? It also have the ability to fix nitrogen in specialized cell. They also have the specialized cell. And though these cells that are known as heterocysts, they are known as heterocysts. So now we talk that how bacteria do nitrogen fixation. How bacteria do nitrogen fixation. How bacteria do nitrogen fixation. These bacteria that fix the nitrogen, they basically have the special enzymes. They have the enzymes that are known as nitrogenase. They have the nitrogenase enzyme. Nitrogen made up of two subunits. Basically, nitrogen contain two subunits. They are, one is the molybdenum and other is the iron. They contain these two subunits, and this enzyme work in anaerobic condition This nitrogenase enzyme works in anaerobic condition. This works in anaerobic condition. Nitrogen fixing bacteria are basically anaerobic. So nitrogenases are rapidly degraded by oxygen. For this reason, many bacteria cease production of the enzymes in the presence of the oxygen, and many nitrogen fixing organisms exist only in anaerobic condition, respiring to draw down oxygen level or binding the oxygen with a protein such as lag haemoglobin. But for example, Rhizobium, they survive in aerobic condition. To do nitrogen fixation, they have to convert into an aerobic condition. So in this enzyme nitrogenase, there is a compound that are called lag haemoglobin, that are lag haemoglobin. So it absorbs all the oxygen and excrete out, so that it can again become anaerobic. When they are in aerobic condition, they cease their production. So this lag haemoglobin, it absorbs the oxygen and excrete out. That then it can become the anaerobic. So now we talk the free living state. In the free living state, and second are the symbiotic state. First we talk about the free living state. Free living state. So what happened in the free living state? That the example nostoc. Nostoc. It is a type of cyanobacteria. It is the type of cyanobacteria, and second, the structure of nostoc is like filamentous, and nitrogen fixation in nostoc can be done through special structure called heterocysts, and that it has special structure that are heterocysts. These heterocysts basically like H-shaped structure, and heterocysts all help them in conversion of aerobic condition and by making their cell wall thick. So what happened? Heterocysts all them helps that in conversion of anaerobic condition by making their cell wall thick. They make their cell wall they become thick. So in other hand, we talk about a symbiotic state. Symbiotic state. Symbiotic state. In symbiotic state, there is a rhizobium. Rhizobium. Rhizobium are bacteria present in soil and around the root hairs of plant. This bacteria basically lives in the roots of the plant. Lives in roots of plant. So they form chain and enter into the roots hair. Rhizobium make the root hairs curl by their properties. They make the roots curl by their because it has the properties. That's why next they move to the cortex region and made the tumor-like structure and excess growth of cortical cells. And this clump of cell is known as nodule formation, and in this clump bacteria are present. So the symbiotic state, the rhizobium, are the bacteria that are working. So now we talk about the mechanism, how this the mechanism take place. Mechanism. In the mechanism, bacteria have nitrogenase enzyme, and that nitrogenase it has basically triple bond, right? There are some compound required for the nitrogen fixation. These compounds are in which the one is the nitrogenase enzyme, other is the lag hemoglobin, and third are the hydrogenase. So these three compounds required for the nitrogen fixation. So this hydrogenase enzymes react with the nitrogenase. First of all, what happened? This hydrogenase, when react to the nitrogenase, it can degrade, it can break this bond, this triple bond, break into double bond. So now we have two bonds, right? So what happened? In next step, the hydrogenase enzyme again break it and it converts into the single bond. So it can convert into the single bond, and finally it again reacts to the nitrogenase, and it also break this single bond. And what happened? This hydrogenase enzyme bind to this nitrogen, so it make the ammonia. It form the NH3. So this is the mechanism behind the nitrogen fixation. So nitrogen molecule break and bind to the hydrogen, so NH3 is made. So this is all about the nitrogen fixation. If you like this video, like, share and subscribe it. Thank you.